risk modeling of complex phenotypes improves risk prediction? Please.
Yeah, so hello everyone. Thank you for coming to my talk. So today I'm going to be explaining how the integration of rare variants in genetic risk modeling of complex phenotype can improve the risk prediction. So a bit of background. This graph shows different categories of alleles depending on their effect size and allele frequency. On the y-axis, you can see the effect size from low to high, and then on the x-axis, you can see the allele frequency from very rare to common. And as we all know, most of the identified rare alleles cause Mendelian diseases. So one allele in one gene can cause one disease, and they have high effect size. On the other side, we have the common variants with low effect size, and their additive effect can contribute to the genetic architecture of complex phenotypes. There are there are few maybe common variants with high effect size that can affect phenotypes, and on the other hand, there are rare variants with very small effect size, but these are hard to identify because the effect can't be detected or is hard to be detected. In the middle, then we have low frequency variants with intermediate effect, and these are not or just been recently studied, and we think that these variants can contribute also to the genetic architecture of complex traits, and this is what we're trying to investigate. So fortunately, Frederica already told you a bit about polygenic risk scores, and basically they estimate the genetic risk of an individual to develop a certain trait or disease, and they are calculated by computing the sum of the risk alleles that correspond to the certain phenotype. And the effect size is usually derived from the GWAS, as Frederica mentioned. On the other hand, the rare variants are usually analyzed using gene-based analysis, and again, Axel mentioned a bit about this, where you collapse the rare variants in a genetic region. Usually, first you identify the damaging variants, as shown in this figure. Once you identify them, you can either give them a binary or a quantitative score, depending on how you want to go around it. So there are multiple ways. And then you basically sum all these scores together into one score, and then you can basically do the association analysis. So our aim is to first analyze the association of rare functional variants in exome region on complex phenotypes, and then integrate the common variants using polygenic risk scores and rare variants using the gene-based scores in genetic risk modeling. To do that, we have developed a pipeline called GenRisk. This pipeline is an open source Python package, so you can find it in this GitHub link. You can also read our recently accepted manuscript in bioinformatics to know more about it. But the main idea of it, or the main work frame, is that we have a VCF as an input data file. This VCF can be used to create polygenic risk scores using BGS catalog API to download the files of the BGS, and then it calculates them for the cohort, or you can use them to calculate the gene-based scores. These gene-based scores use functional annotations like CAD or Revel, so quantitative deleterious score, and the allele frequency. In these scores, we use a functional weight, so that the rarer the variant, the higher the score. This way, it basically upweights the rare variants. And then you end up with a matrix of gene-based scores. This matrix can be used with the phenotypes either to do association analysis using the linear regression or logistic regression, or we can use the matrix with the phenotype and the polygenic risk scores to generate prediction models. So for our cohort, we used UK Biobank data. We started with 200,000 samples. We filtered for Caucasian and British to end up with 160,000 samples. We calculated the gene-based scores for these samples with variance of minor allele frequency of less than 1% using the exome data. And then we basically analyzed the phenotype, as phenotypes, the biomarkers from UK Biobank. These biomarkers are quantitative, and we basically used this paper here shown as a reference. We calculated the BRS default by the same paper here, and then but when we calculated this BRS, we excluded the variants that are present in this in the gene-based scores from the calculations, just to avoid collinearity. So the gene-based scores independently don't have enough power to be used as features in the prediction models. So what we did is we did something similar to polygenic risk score for the gene-based risk score, for the gene-based scores, by creating a gene-based risk scores. And we basically took 50,000 samples from our main cohort, and we did association analysis on it. Using the statistics from this association analysis, we created an effect size by using the beta coefficient divided by the standard error, and then we multiplied that by the gene score and added all these effects together to create the gene-based risk score. So then we created prediction models for each biomarker. Each biomarker had the same covariates, so age, sex, BMI, and the first four genetic principal components. And we generated 3 different models. One was with the BRS and the covariates, one with the gene-based risk score and the covariates, and then one combined model with BRS, GBRS, and the covariates. We used 75% of the data for training with 10-fold cross-validation, and then we use 25%, the rest of the 25% for the independent testing. And then we run this on multiple regression models and selected the model with the best performance. So for the results, let's start first with the association analysis. So as I said, we use gene-based risk score and the phenotype to run association analysis. And here are some results for the association analysis. We used linear regression, and then we corrected using FDR. And this table shows the top 3 significant gene associations. So most of these associations have literature evidence related to the corresponding phenotype, but I will only mention for now the cholesterol. Also, I need to mention that for the cholesterol, we had to adjust for people who were taking statin as medication to raise the cholesterol level to accommodate for that. So all the 3 genes here are previously known to be associated with cholesterol. For example, the inhibition of PCSK9 reduces plasma LDL levels. The LDL receptor orchestrates cholesterol homeostasis, and the ABCA1 is the primary gatekeeper for eliminating tissue cholesterol. So next we went for the prediction models, and as a reminder, we used the gene-based risk scores with the polygenic risk scores and the phenotypic data to create the prediction models. So you don't really need to look deeply into this graph because it's too big, but basically we have as blue the GBRS and the BRS models, so the combined models. The orange is the BRS model, and then the green is the GBRS model. And this graph shows that most combined models show improved predictions. So 25 out of the 32 biomarkers show improvement when combining the BRS with the GBRS. Let's take a closer look to a couple of examples. So here we can see that, for example, aspartate aminotransferase, cholesterol, and alkaline phosphatase show improvement when using the combined model rather than the BRS individually or the GBRS individually, while LDL direct and direct bilirubin don't show improvement. And this just to mention that the contribution of the GBRS depends really on the phenotype and the genetic architecture of that phenotype. So I hope with this I convinced you that rare variants contribute to the genetic architecture of complex traits, and that the integration of rare variants in genetic risk analysis of complex phenotypes can improve risk predictions. For future work, we would like to apply GenRisk pipeline on different traits and populations. So if you're interested, please contact us. And we would like to do also pathway analysis and enrichment for our results. Thank you for listening.
Are there any questions to this related to this talk? Well, thanks first for a nice presentation. I cannot see one. You said that you separated your data set into the first part with 75% and another of 25.
Yeah.
Why don't you do half and half?
Half and half. I mean, I never thought about doing half and half. Really, the idea is that we have enough samples to use most of it for training, and then have enough to have a independent testing sample that shows us either improvement or not. So you don't like it wouldn't affect. I mean, it would be better always to have more training set, and since we have enough, like it was 25,000 maybe for testing, which is enough to say that if there's an improvement in the prediction or not.
Okay, thank you. Is there another question? Probably related to this time of the day, so I think. Thank you.
Yeah, thank you very